And good afternoon, everyone. So, yes, and welcome to Research Grand Rounds. Really great to have an opportunity to share research ideas and see how we are doing and where we are going. So it's my one of my favorite conferences here, not the most favorite. We're very pleased today to hear from Dr. Ratajczak, who is in our stem cell program in the Division of Hematology here in the Department of Medicine. He's going to speak on from a concept of stem cell plasticity to discovery of a very small embryonic-like stem cells, VSELs. Way cool. So wait. I have to brag about you for a little bit first. Okay. So Dr. Ratajczak is actually originally from Poland, and he actually obtained his Doctor of Science in Warsaw. His Ph.D. also came from the Polish Academy of Sciences in Warsaw, and his M.D. From Szczecin in Poland, and where he was magna cum laude, valedictorian in among 280 students. Congratulations on that. And he actually came to us after a lot of training in Poland and in the United States at Penn. And he's been a prolific author and researcher, highly funded, and more important, very impactful. And so thank you for all that you have done and continue to do. And we're looking forward to what you have to tell us today.
Yeah. I would like to thank very much for invitation to this meeting and this kind of introduction from the two Williams and also Jason for preparing this meeting. And I will be talking today about regenerative medicine. I'm working 45 years already in science area. And also very small embryonic-like stem cells we discovered. And university was awarded 2 years ago patents. Not submitted, but awarded patents on the cells. Yeah, and now is the question, what do we do next? And before I will start, I am always showing this slide because it is important. We will be challenging some dogmas, and we should beware of dogmas. And how Albert Einstein said, blind belief in authority is greatest enemy of truth. And Max Planck, also said one rule is important in science that only courageous people can win. We'll be talking first about stem cells, we can employ right now, which are on the market in regenerative medicine, and later about some strategies. And as you know, unfortunately, there's here a lot of conflict of interest. Patents, companies, private investigators, and still we don't know which cells we'll use in the future. We need to have more important stem cells. This means cells which will differentiate in all three germ layers: meso, ecto, and endoderm. And the question: embryonic, induced, or adult tissue-derived pluripotent stem cells. And just very briefly, as is shown here, two different strategies to get cells from embryos. Normal fertilization, which is employed in the clinic, in vitro fertilization clinic. Some extra embryos are frozen. You can, from zygote, have morula, and from blastocysts, isolate theoretically stem cells. It's not so easy. It is why we don't have so many stem cells from different reasons, ethical, first of all, on the market. But these stem cells will be histocompatible only with the parents, because they will have histocompatibility genes from sperm and oocyte. Another strategy, therapeutic cloning, where we take oocyte, remove nucleus, and instead, in place of this nucleus, we put nucleus from a patient cell. And of course, then we can make customized histocompatible stem cells. And what is here important is an evidence that clonot, like zygote, has a lot of microRNAs, proteins, which are working on this DNA and make from this DNA virgin DNA, as we see in embryonic stem cells. And again, we get morula and we can get stem cells. Another strategy, it is to, by genetic induction, that we can, it also not so easy process, but to have a plasmid with so-called Yamanaka factors, we put to somatic cell, and if we are very lucky, several thousand of cells are exposed to such plasmid. Sometimes we see growing clones. People also were trying, instead of plasmid, to use microRNA, messenger RNA, and even proteins. But are problems with these cells. Even if many grants are awarded and a lot of people are working, we have to look for this first. I have to show with finger because it's not working, unfortunately. These all cells isolated from by fertilization, by therapeutic cloning, and induced pluripotent stem cells grow teratomas. This means, it is very hard to predict if they will ever will employed in the clinic because of this problem. It is why a question is, where is going regenerative medicine? And is first question, if pluripotent stem cells, which differentiate in all three germ layers, are present in adult tissues. And I show you our journey, from concept of stem cell plasticity, which we never believed, to discovery of VSELs. And as you know, it is already the slide, 23 years old from Nature, where it is shown that, marrow cells, hematopoietic mostly, can differentiate in all these tissues shown on the slide. But, nobody was later able to repeat this data. Nobody retracted this paper published in Cell in Science and Nature. And from the beginning, it was already 20 years ago, we were very skeptic. And we were showing, okay, if we are using cells from the bone marrow or cord blood, then are different here effects. And first, we propose that stem cells can secrete growth factors, cytokines, and chemokines. And actually, this paper published 25 years ago was first paper in literature showing that CD34 positive cells, this means hematopoietic stem cells, secrete several, at protein level, several chemokines, growth factors, which stimulate hematopoiesis, inhibit apoptosis, stimulate vasculogenesis. It was first paper showing at protein level. Then another concept, which we consider, will biological effects of stem cell-derived microvesicles. It is interesting because, it was also first paper in literature, 3,000 times right now cited. And now everybody is doing microparticles, but you can believe me that, when we presented this data first at the American Society of Hematology in 2002, it took 4 years to publish this paper. And after this paper showing transfer of messenger RNA, microRNA by microparticles, people jumped in this area, and right now several thousand of papers are published, and many laboratories are working with microparticles. And also, to show that this paper here from Louisville was first, it was a commentary in PNAS. And as you can see here, they acknowledge that we were first in the literature showing this phenomenon. This means if we look because we are interested in heart regeneration, personally, I don't believe so much that mesenchymal cells, hematopoietic cells, doing, new cardiomyocytes. This what we see is secretome from these cells, microparticles, and also soluble factors, which prevent damage of myocardium. And we have also proposed already, many years ago, that we can engineer these microvesicles by enriching messenger RNA proteins and use in the clinic. And also coming to induced pluripotent stem cells. Also, this paper is published already, almost 15 years back. We told at the time that actually induced pluripotent stem cells don't have much application unless they will be used as a source of microvesicles. And and Japanese people, because Yamanaka got a Nobel Prize for induced pluripotent stem cells, they were doing a big clinical trial in cardiology using cells derived from induced pluripotent stem cells. And conclusion of this very big trial was that if anything is working, then secretome from induced pluripotent stem cells secreted microvesicles and growth factors. And third, explanation was that maybe what we expected at the time, among normal bone marrow cells, cord blood cells, we have some real diamonds, very rare cells, which can differentiate across germ layer. And to show you, here what people believe for many years, that embryo which have pluripotent stem cells in inner cell mass of blastocysts is giving rise to tissue-committed stem cells, and these pluripotent stem cells disappear. And we challenge this, that maybe it's not true. Maybe these pluripotent stem cells still survive in adult tissues as a backup population, but they have to be quiescent and locked in, because otherwise they would grow teratomas. And teratomas are very rare tumors in humans, yeah? And I will come later to this mechanism. And our two papers which we have published describing these very small cells in murine bone marrow, human cord blood, later human bone marrow, and actually opened a new area of investigation. And here is a electron microscopy pictures, seeing is believing, of very small embryonic-like stem cells, and in comparison, hematopoietic stem cells. Hematopoietic stem cells, from definition, are small lymphocytes. VSELs is much smaller, have very primitive chromatin, and as you can see here, express at protein level stage-specific antigen. One of 4 in nucleus nano. They have normal number of the chromosomes and viable cells because they exclude 7-ADD. And looking, at the time by bulk messenger RNA analysis, we have found that V cells, similarly, there are murine cells, murine V cells, similarly, murine embryonic stem cell line ESD, they have very similar signature of gene expression in contrast to hematopoietic and mononuclear cells. And what is very important, I will come back to this, they express not only genes of pluripotency at blast, but germline specification and germline development. And we have found such cells not only in bone marrow, but also in several adult tissues, in lung, brain, kidney, pancreas. And what is important, number of the cells in the bone marrow, urine data, decreases with the age of animals. Humans accept. And these are human V cells. Also, electron microscopy of these cells isolated as CD133 lineage negative, CD45 negative cells from cord blood, and also mobilized peripheral blood, young adults. And here I make this circle to show you they are not granules around mitochondria. If you make hyperbole, and round mitochondria are very characteristic for germline. This means not only expression of germline-like cells, markers, but also round mitochondria. And as you can see, there are right now over 300 papers, 50 independent laboratories worldwide, independent groups confirmed presence of these cells. We have also postulated that these cells could be mobilized from tissue circulate during tissue organ damage, but of course, it's not a high number of these cells. They try to do something good to regenerate damaged tissues. And also, here's our examples for cardiology. This is important. We have shown here in collaboration with Dr. Boli and also with my collaborators in Poland that in mice and in humans that are released during heart infarct, circulate in peripheral blood at high level, and also in stroke. But not only in stroke, but also in skin burns, in intestine, in Crohn's disease, and even in psychotic syndromes, because we know right now that in psychotic syndromes is some rebuild of brain structure, and maybe stem cells play here a role. And it was the first patent, is already awarded 2 years ago for University of Louisville, not submitted, awarded, on the cells that they exist in adult tissues. And discovery of V cells put new life on tissue organ rejuvenation, aging, adult stem cell therapy, and also malignancies. And now, I show you this developmental link between primordial germ cells, B cells, and hematopoietic stem cells. And here is big credit to Janina, because when we isolated V cells, we were isolating this as CD45 negative cells. Hematopoietic cells are CD45 positive, but these cells were CD45 negative. She said, no, we will try, maybe we can get hematopoietic cells from this. And we use exactly the same system like people were using to differentiate embryonic stem cells to expand these cells over OP9 stroma cells. And we have reported that we can get from these cells stem cells from murine bone marrow and also human cord. And now, because there are also some hematologists here, I remember when I was a student, for me it was a puzzle. Why it was like this, in you were studying embryology, that first hematopoietic stem cells are identified in extraembryonic tissue, not in embryo, in extraembryonic tissue. And it is explanation, first, these green cells are primordial germ cells, are first cells specified in epiblast. What they do? They leave epiblast, go to bottom of yolk sac, then make a turn through primitive streak, go to aorta gonadomesonephros region. And now, please look. First hematopoietic stem cells, as you remember, are identified at the bottom of yolk sac. It is overlap here, migration of the cells, first hematopoietic cells, and then definitive hematopoietic cells are in dorsal aorta, in AGM region. And we have proposed that it is a link between primordial germ cells, V cells, and hematopoietic lineage. And as you can see here also, there were first papers which in very extensive way have shown that hematopoietic cells, murine and human, express several gonadal and pituitary hormone receptors. And here is in vivo experiment. We took mice. Mice were injected with different sex hormones, FSH, LH, danazol, estrogen. And we were looking for, and later, 10 days after this, we were giving mice bromodoxyuridine. Bromodoxyuridine accumulates in cells which amplify DNA, which proliferate. It is a marker of proliferation. And as you can see here, these cells, which are very quiescent, after all these sex hormones, very nicely responded. They started to expand in vivo, in bone marrow. The same, of course, would be expected for hematopoietic stem cells. I am showing this here because later we use this observation to develop ex vivo expansion of these cells for clinical purposes. I show you later. And of course, this our discovery was confirmed later by Massimo De Felici, who is guru in Italy in stem cells and germ cells. This means what we believe, this very primitive cells from germline during development are located in different developing organs, and they are kept quiescent because it's some mechanism which regulate the quiescence. I will tell you later about this mechanism. Now, erasure of parental imprinting will answer why these cells are kept quiescent in adult tissues. What is this? This mechanism of parental imprinting is very well known to embryologists, but unfortunately, many people who are even excellent in genetics don't go back, and don't look, for this mechanism. Here, everything what is shown red is germline. Zygote develops from two germ cells, oocyte sperm, and then through morula inner cell mass of blastocysts to epiblast and to primordial germ cells which migrate to genital ridges to make new gametes, we have everything germline. We also believe that at this point in epiblast, some of cells are located in developing meso, ecto, and endoderm, and these are V cells which have many genes from germline. And Dr. Resnick, already it is 25 years back, have described a mechanism why primordial germ cells which migrate to genital ridges are quiescent in other tissues because otherwise they would grow teratomas. And it was erasure of somatic imprint. And now I will show you how it works. It is shown here zygote, here, let's say, two gametes. And as you can see in gametes, there are around 50 genes, and some are very important, that are differently methylated if cell is coming, is a gamete female or gamete male. And here, for example, this one gene is methylated in sperm, not methylated in oocyte. Vice versa, this one is methylated in oocytes, not methylated in sperm. But if they will come together after fertilization, then we have expression of these genes on reciprocal genes. And such, imprint is in all somatic cells. But is exception of this rule, primordial germ cells. When they migrate to genital ridges, they erase imprinting on precursor of gametes, and this imprinting has again to be re-established on gametes before fertilization. And we have postulated the same mechanism happens in VSELs. And now to have a proof that it is true, we looked for these imprinted genes. And you would be surprised because many of these imprinted genes, the most important, regulate insulin, insulin-like growth factor, somatotropic signaling. Shown here in the red, these most important genes. And they are important for cell proliferation and metabolism. And example of one gene which is very important. It is a tandem gene, insulin growth factor 2, H19. This is a growth factor. H19 is just cut to several microRNAs, and this microRNAs have inhibitory effect on cell proliferation. And as you can see here, what happened? Maternal chromosome, paternal chromosome. These two genes are separated here by imprinted sequences. When it is here imprinting, methylation, then CTCF, insulator, cannot bind to these sequences. But on maternal chromosome is erasure, right? And CTCF binds. And what is a consequence? It is a consequence that this CTCF make on maternal chromosome a border between H19 and IGF2, and on paternal chromosome, distal enhancer regulate IGF2 from paternal chromosome. And what is a result of this? A result of this is that, if we have erasure of imprint, because it is normal situation, but if we have erasure of imprint and to both here regulatory differentially methylated region binds insulator CTCF, then distal enhancer stimulates expression of inhibitory H19 from both chromosomes, paternal and maternal. Cells have a lot of H19, very little of IGF2. And this happened in primordial germ cells, and we have shown this happened in our V cells. And to show this at the molecular level, you can look for methylation, if you have primers, for this loci, and you make bisulfide modification followed by sequencing. You can see that V cells have erased all these loci which are not methylated, in contrast to hematopoietic cells, which is 50-50%, because maternal and paternal chromosome are okay. Looking at messenger RNA expression, these cells have a lot of H19, which inhibits proliferation, and they have relatively low level of IGF2, which promotes proliferation. And now, we were aware that we have to reverse this phenomenon. We have again to methylate one of these, chromosomes. And in order to methylate this, it is a job of de novo methylotransferases. And we have three de novo methylotransferases, 3A, 3B, and 3L. And they work this way that, they need, in both cases, the de novo methylase 3L to remethylate one of these chromosomes. And we have found that VSELs have a very high expression of this de novo methylase 3L. And what happened here? De novo methylase 3L is chaperoned by sirtuin. And now, to release this as free protein, which could bind to 3A and 3B, we need some compound. What is this compound? Nicotinamide. Nicotinamide is blocking this interaction and releases DNA methylase 3 for methylation. And nicotinamide, as you know, it is used also as some supplement. This means, in presence of nicotinamide, we have a gain we can have in remethylated, after erasure of somatic imprint, remethylated chromosomes and these differently methylated regions. It was an example of IGF-2H19, but it works similarly with few other genes. And we have described, this phenomenon, a couple of years ago. And based on this, we developed expansion of these cells for practical clinical purposes. As you can see, very small cells here after sorting. And it was always a problem. When we have so very few cells, how we could go for regenerative medicine. But in this experiment here, Janina Oshwer was doing, if we expose these cells to nicotinamide and add combination of some of these sex hormones, like folliculostimulin, luteinizing hormone, danazol, we can expand after 2-3 weeks these cells. And what is important, we have something what is very typical for stem cell. We have here asymmetric divisions. We are getting some larger differentiated cells, but as you will look around, you have still a lot of these small, very small embryonic-like stem cells. And it is a second patent awarded for university 2 years ago on expansion. This means we have right now tool to have this cell for clinical application. It's not submitted, it's already granted to the university. This means if we look, because also some people from hematopoiesis, we want to expand hematopoietic stem cells for clinical purposes because we need more stem cells for successful bone marrow or cord blood transplant. You know that right now is not any reasonable expansion strategy of hematopoietic stem cells. Every year somebody is coming with new combination of growth factors, and every year the same story repeats. We are always, expanding something what are progenitors, and we lose stem cells. This means what we propose for a proper expansion, we can go to B-cell level because these cells can serve, renew, and then going down in this differentiation pathways. Now, few words for aging. And another goal of regenerative medicine to develop strategies to increase the robustness of adult stem cells residing in adult tissue. Sometimes we don't think about this, but it is very important. If we have these cells in our body, we have to make everything to make them happy, that they can be in good shape, and they can regenerate our tissues. And again, we are coming to this scheme, insulin-like growth factor signal. And as you know very well, probably the best way right now to extend longevity is caloric restriction. To cut by 30% our food intake. And what is doing caloric restriction? Down-regulating growth hormone, insulin-like growth factor. And this translates on extension of lifespan and lower risk of cancer. But it's reversible. High caloric diet increases growth hormone level, insulin-like growth factor, and it is decreased lifespan, higher risk of cancer. Best example, obese women, unfortunately, very often develop a lot of tumors, uterus, breast, etc., but also obese men. And if we look in animal world, if we have here yeast, C. elegans, Drosophila melanogaster, or mice, whenever we have decrease in insulin somatotropic signaling, we have extended lifespan. Genes are called differently, because genes which are in mice are a little bit different than in yeast, but the whole idea is the same. Decrease insulin-like growth factor somatotropic signaling, and this results in increase of lifespan. And we did such experiments here in collaboration with Dr. Bartke from University of Springfield, and and we have found that mice he has, they were long-living, short-living mice. These long-living mice can live even up to 4 or 5 years. And these are called the larron-dwarf mice, long-living animals. And we got these mice from him, and we look for number of VSELs, and we have found that these mice have a lot of VSELs in comparison to control mice. And also what is here important, these mice very rarely have tumors. Very rare. In animals, if you have in animal facility, after 2 years, almost every mouse has some problem, develop tumor. These mice don't develop tumors. And also, as you know, one of the first things which fails out in our life is reproduction. And here we have been looking for ovaries, wild-type mice, larron-dwarf mice. And as you can see, these larron-dwarf females have smaller ovaries, but wild-type mice at 2 years have already fibrotic ovaries. But larron-dwarf mice have still active ovulogenesis. And we were getting from this larron-dwarf mice pups at age of 2.5 years, which is impossible, with wild-type mice, because these mice after 1.5 years, usually don't have any offsprings. And it was confirmed also by other, investigators, our collaborators, that, in ovaries, VSELs could be precursors of gametes. And here is a very nice paper from Irma Wieland-Klun from Slovenia. She has isolated, VSELs, as you can see here. Then, she differentiated them to oocyte-like cells. And then what is a very important evidence, nobody was able to repeat this with any type of stem cells. In presence of sperm, they release zona pellucida. It's the first step of fertilization and very nice evidence that these are functional oocytes. If we will use these cells in future for fertilization, we don't know, but such phenomenon exists. Now, coming back to this decrease of VSELs in normal mice with age, from 2 months to 36 months, you can see here. What is here very important, that we have thing to extend lifespan by preventing rapid decrease of the cells in adult tissue. And we can do different things. Not only caloric restriction, what we did here, in mice, people are doing this, but we have shown that this correlates with that we can save this way number of VSELs in bone marrow in adult tissues. But also endurance exercise is very good here. We were trained mice on treadmills. They were running every day. Mice can run to 5-6 kilometers in 1 hour. Are really very good, runners. Yeah, but we didn't run them every day for 5 kilometers. But but also this increased number of very small embryonic-like stem cells in adult tissues. And also, what is here very important, what we have postulated, is that prolonged growth hormone, insulin-like growth factor signaling, is silent killer of stem cells and culprit in aging in our adult tissue. Many young men, take growth hormone. Because build masses, etc. But it is two-edged sword. After a time, you can deplete VSELs from your tissues. And it is why what we postulate that very small embryonic-like stem cells, these cells which are responsible for regeneration, which are on top of stem cell hierarchy, are burned during our life in a metabolic fire of insulin-like growth factor signaling. Now, a few words, still I have a couple of minutes for cancerogenesis. Levi will be interested here. Same here, pathways. And, it is like this that I was studying medicine back in Europe. And in Europe, it was very, very popular, hypothesis embryonal risk theory of cancer development. is coming back to Rudolf Virchow, Julius Konheim, and John Beard. It was like, 19th century. Nobody was talking at this time about stem cells. But, Rudolf Virchow, he had, some vision that maybe some of the tumors in our body, especially anaplastic tumors, develop from some embryonic remnants. And we think that the discovery of VSELs brings this great idea, alive. And, what is also the supporting theory of cancer development in germ cell compartment? Presence of classical germ tumors, from gametes, from mediastin to the brain, rare, but you can have germs tumors like this. Expression of cancer testis antigens. Many tumors, like even lung cancer, bladder cancer, all type of tumors, they express cancer testis antigens. We can ask, okay, why? Maybe because, in tumor everything is expressed, or maybe some of these tumors derive from germline. Expression of choriogonadotropin and carcinoembryonic antigen. Now, maybe it's not so very famous, but a couple of years ago, when I was still, more active, they were used in the clinic to detect potential malignancy. These are embryonic markers. And expression of OCT4 in several tumors, it is reported in several tumors. And, if you look in our VSELs, they express here at very high level selected, shown here, cancer testis antigens. And now, something which is relevant to pediatric sarcomas. As you look here, it is a proper somatic imprint, balance between IGF-2, H19. But we are talking about erasure of somatic, but it is also loss of imprint. Loss of imprint means all loci here, regulatory loci, are methylated. What is the consequence? Distal enhancer regulates expression of IGF-2. These cells have a lot of IGF-2. And we see this in Beckwith-Wiedemann syndrome, but also in most of the pediatric sarcomas, in rhabdomyosarcoma, neuroblastoma. We see, increase in this mechanism. And we believe, because these are tumors which are young patients usually, that these tumors, sarcomas, are derived from VSELs. And also here, acute leukemia. Several examples here, as you can see, that the OCT4 nano genes expressed in leukemia blast. And there are three different scenarios, what we postulate, how VSELs could contribute to malignancies. First, if some imprinting, persistent imprinting, they can grow teratomas. If mutations, they grow also tumor. Then VSELs can just sense growing tumors as damaged organs because it's hypoxia. And they home to this growing tumor and provide stroma and vasculature. Because, they are cheated here by tumor, calling I'm hypoxic and, are contributing to better vascularization. And also they can fuse with cells, normal cells, and give aneuploid tumors. And at the end, we are talking about mice, but it is such mutation in humans around the world. It is very interesting. This mutation is coming mostly by Sephardic Jews. A group of this patient is living right now in Israel. And many of these patients are in Ecuador because they ran away from Inquisition like 300 years back. And a cohort of this patient is in Ecuador, a patient is in Israel. And the Laron mutation is even, named after Dr. Zvi Laron, who describes this mutation. And this mutation is lack of circulating IGF-1. This means no IGF-1 circulating in peripheral blood. You have, of course, decrease in growth, but please look here. There are right now 400 cases of Laron-Dwarf reported very well. As you can see, in brother and sisters, because you have to be homozygote, you have like 20-25% of cancer. No one cancer so far was described in Laron-Dwarfs. It is amazing, yeah, that only such one mutation and they are resistant to cancer, like mice. Mice also have very rare mutation. Because why? They don't have circulating IGF-1. Mice don't have circulating IGF-1. Patients don't have circulating IGF-1. But you can ask, okay, do they live long? No, it is complicated. Why complicated? Because unfortunately, are poor individuals, and there are other causes of death. And also, what is interesting, they almost don't have diabetes and don't have a problem with heart. This is not worse. Yeah, but as you can see here, why they are dying? Accidents. Are not accidents, are suicides. Because they are unhappy, Second, alcohol-related and drug-related problems. And then, most what is convulsive disorders, it is a problem because they have changed, the school, and they have a lot of epilepsy. This means to summarize this, my lecture, in order to have balance between VSELs and insulin somatotropic signaling, if we have this balance in steady state condition, everything is perfect. If insulin signaling prevails, we have accelerated senescence cancerogenesis. Vice versa, when we have here low insulin factor signaling because of diet, because of exercise, caloric restriction is more VSELs, and we have prolonged longevity, lower risk of cancerogenesis. And just few slides to show you what we are working right now, what is relevant to today's pathology. We have shown that not only stem cells, but VSELs express a receptor for SARS, for COVID-19 entry. And now, it raises a question, we know how it is with herpes virus, survives in nerves. We have this all long COVID problems. Now, it's very likely that COVID survives in stem cell compartment and could be activated if it's not under control, and people have this long COVID problems. Another, remember last year I was showing about cholinergic signaling, and I was showing a lecture about complosome, intracellular complement. And what is very interesting, VSELs express all receptor for cholinergic signaling, respond by chemotaxis to ATP, and also VSELs express complosome proteins. This is a new area of investigation. And finally, many of this data was done by bulk RNA. But we want to do this at single cell level. And we have repeated this data with VSELs, sorted VSELs, and make single cell seq RNA analysis. And there are here what we have found. We confirmed that we have found genes annotated to germline compartment, regulated by parental imprinting, responding to early developmental phase decision, and transcription factors involved in early development. And conclusions for today, the quest for pluripotent stem cells, which could be employed in the clinic, is still going on. I don't want to offend anybody, but as I told, I don't believe, and I wish, that induced pluripotent stem cells, or embryonic stem cells will find application in the clinic. Teratoma formation is a huge problem, and nobody, after so many years of discovery of the cells can omit this. Right now, most of stem cell therapies effects we have in the clinic are paracrine effects by secreted microvesicles and by secreted growth factors and other mediators. But evidence accumulated that adult tissue contain developmental early stem cells, and our new VSEL expansion strategy, right now supported by patent, make a hope maybe for employing them in clinical settings. And most important, people who work in regenerative medicine have also to understand that we have to, what I mentioned, to make our stem cells happy. Because if they are happy, we will rejuvenate our tissue, we will be in much better condition. And here is a lot of improvement, for some pharmacological drugs. Metformin is good, but we need more specific drugs than metformin to modify insulin signaling. But also exercise, and caloric restriction. And here is web page. You can go and check on this. And I would like to thank very much. First of all, now back in Poland, Professor Magdalena Kucia, she's right now in Warsaw. Janina is here in Louisville, and other collaborators. And what is interesting at the end, a friend of mine who is in Poland, Speaker of the Senate, very recently published a book. And it is unfortunately in Polish. But in this book, he describes, like 1/4 of this book describes our problems we had here to fight our competitors in Louisville, who, didn't like our cells because, they were afraid that maybe some company will come tell, okay, we don't use this cells, we will use your cells, And this is, a book he wrote, and yeah, it will be for sure translated, but it is interesting because its action is here, in Louisville, in our center, which is, invaded by some people from Vatican even, because they don't like sensors. and he had imaginations that from this small building is some walkway underground to the airport, etc. Anyway, thank you so much.
Well, thank you so much. This was very insightful, and I have to compliment you on breaking with Max. You showed Max Planck, but the Max Planck principle is that science doesn't advance by doing something incredible like you did, and then convincing the existing dogma to accept it.
Yes.
They all have to die off. And then it becomes accepted by new people who have an open mind. Somehow, in a matter of years, you actually got everyone on board, which was pretty amazing.
Yeah.
So
definitely, we were. It is a very complicated story because I don't want to go in details, but our technology office here, did not the best job, looking for investors, and they found investors, and it was wrong people. Yeah, but anyway, still open area of investigation, and maybe some company will come sooner or later, and we can go forward.
Right.
As well.
So that was my comment. My real question was, you have vessels, you have stroma. What's next? Can you do, glomeruli and tubules and make not just a kidney, but a sheet that has renal function? So the kidney function would actually do that. The heart, you have to actually get everything together in one place to make it into a pump. But I filter like a kidney.
Yeah, it's doing, regeneration of large organs and tissues is a huge problem.
Yeah.
Because it's not only to have, a ball of cardiomyocytes. Let's say you need to have vessels,
Right.
you have to have lymphatics, you have to have nerves and everything. And also, you have to also have, proper activation of this everything, And it is a huge problem right now. People are doing some small organoids, samples of the tissue, but, to grow a real heart or organ, it is huge challenge. It is huge challenges for now, but maybe we will go to Mars. We will grow also.
And that's why I was thinking not a heart, but a kidney, where
kidney, of course.
You could replace, you don't need the organ just like a sheet of functioning tissue that could actually do the job.
Yeah, no, definitely. We have also found VSELs in kidney, as well. Yeah.
Right.
In all organs.
Other questions?
So I have a couple questions. So the obvious one is, so if you give VSELs to mice, do they help them live longer?
Yeah, it is a very good question. And I tell you maybe a reverse experiment. Yeah, when we give to mice, which are long-living mice, insulin, we shorten lifespan. If we overexpress growth hormone, also they are living only half a year. Not 2 years. So it's mean in other direction we see, and it is collected with decrease of VSELs. Yeah.
And then as far as the experiment you showed with the expansion of VSELs and the nicotinamide.
Yeah.
So does that, when you restore that imprinting, does that cause the pluripotency of the V cells to go away, or?
No. Because, I was showing this one of the slides from expansions that were little cells
Mhm.
and larger. This means they are going asymmetric division. Still, these little ones, they have pluripotency markers. For how long we can go, it depends everything, because doing experiments like this, we need more hands, and etc. Mostly this expansion was done by Janina, and we kept these cultures for 2-3 months, but we don't know how long we can expand. But anyway, patent is awarded on expansion. If company will come here with some money, and we will have a real possibility to have GMP grade cells expand, we can try.
Yeah, so you showed that calorie restriction in
Yeah.
mice makes smaller mice compared to the wild type, and they live longer, their reproductive cycle is extended.
Yes, Shama.
But I did not pay attention being reproductive biologist. Is there any published data about women who are skinny and comparatively shorter than the regular one? Do they have
I look up to this because what
longer
we were showing,
cycle?
what we were showing is that caloric restriction, of course, extends lifespan. People will have reported this, not
Yeah.
first,
Yeah.
but we have correlated this with number of VSELs. Living longer, they have VSELs, and now we know about this. It is something what is good for me, maybe not good for Levi. There were studies in France comparing short and tall men.
So, men or women?
And also, athletes.
Yeah.
That, high, and it was negative correlation. That short, like jockey, for example, are living much longer. Yeah, but of course, in humans, we have a lot of pathologies, lipid pathology, etc. But it was a study done in France and it was in Scandinavia, showing that short men are living longer than taller men. It's not popular studies, but of course it is. We should, be very cautious about these observations. Yeah?
But
Because why? Because if you are growing, the same with dogs. You
yes,
see, little dogs are living very long. Chihuahua, Pomeranian, up to 18 years.
Mhm.
But Bernardines, 6-7 years. Yeah. This means, if it's a lot of stimulation of growth hormone and insulin is not always good. Yeah.
So I have a question. So we are talking about intra-species, but if you look at it, interspecies in mammalian group,
Those, I am not feeling very well.
so
My wife is happy because she knows this.
we are talking about population intra-species. But if you look at interspecies, those, at least in mammalian group, those which are larger, they tend to live longer.
Yeah.
So that's my first one.
Very, very good question. We know because we didn't do this experiment in dogs. Dogs would be very nice model, but
And, also these are like artificial environments. So if you look for, open environment, wild, most intra-species changes, the longevity are not there.
I think that, coming back, also to this, your questions, this phenomenon, of these cells, maybe it was very early in evolution. Because if you look, for example, in urodels, you have something like what is called, this very primitive population of stem cells, which accumulate in place of tissue injury, and they regrow, amputated tail, amputated lens. This means it happens something in evolution. Of course, later on in humans, we see, for example, that after heart infarct, after stroke, after several complications, we see increase of the cells circulating in peripheral blood. This means they want to do something, but there are not too many. But we believe that, for example, some minor tissue injuries, like after marathon run, marathon runner, we have a lot of micro damages in tissues. Yeah, they may play a role, in regeneration. And in fact, we were doing experiments in marathon runner. They have, after marathon run, very high level of circulating V-cells in peripheral blood, other stem cells as well, but also V-cells.
Yeah, I have another question. So again, this is why don't you use like a formulatory secretome instead of live cells which have their own complications?
No, I tell you, we initiated, this, and it's true. You can check. We initiated this, whole story with microparticles in literature. Later, we stopped doing this so much because we started to work with V-cells, and you cannot do everything. Of course, it's a good question. Why not to use cells? Why to secretome? But definitely, if you are using mesenchymal stem cells, they will never grow hard. Never.
Exactly.
And these are data, they will just perish. You inject, after 2 months, you don't see any. But what they are doing, they secrete a lot of factors and microvesicles. And if we see any beneficial effect, it is secretome. Of course, now question, maybe better to use secretome. I don't know.
And that is targeted.
Yeah, but it needs studies, and yeah.
Okay.
Doctor, we had a question from Dr. Kiri is online, would like to ask you a question. Go ahead.
You can unmute.
Hello. Good afternoon. So first of all, it was a wonderful talk, and this is something that we all have been reading about, but there's not much evidence to go about. So I had a couple of questions. Firstly, in oncology, the first concern that we have is the tumor genesis. So what are the potential, like how, if we are going ahead with using the VSELs, so there's always a potential risk of having a tumorigenesis effect. So how do we answer that? How do we deal with that? And
yeah, I have a very big request. Now, what happened here, Dr. Kersti? Now I can hear you because it was, mutated microphone. Please repeat the question, please.
Okay. That's all right. Can you hear me clearly now?
Yeah.
Okay. So I said that one of the concerns we have in oncology always would be the tumorigenesis effect that these VSELs might have. So proceeding on further in the future, when we do conduct trials related to this, and we do plan to integrate it in the human subjects, won't this be a concern? And if it is, then how do we safeguard it? Like, how do we make sure that these don't convert into the tumors that we're actually preventing it from?
Yeah, it's a very good question. I tell you, I am not working so directly in tumorigenesis. It was most hypothesis, which right now is continued by Dr. Deepa Bhartiya from our collaborator, from India. And she has a lot of evidence, that tumors can develop in V-cell compartment. Because we were mostly interested, V-cells and hematopoietic cells, V-cells and hematopoietic cell expansion. But she did a lot of very nice work. You can go to PubMed and check Deepa Bhartiya from India, and she has a very nice papers, confirming that probably, V-cells really contribute to tumorigenesis directly when they mutate. And also in growing tumor, what people were always postulating mesenchymal epithelial transition, that V-cells maybe are here responsible because they recognize hypoxic tumor as damaged organ. They're cheated here. And they migrate to this tumor and provide stroma and provide vasculature.
Okay, thank you. And actually, I've had the pleasure of working with Dr. Vinita from India. She was one of the superiors I was working with on a couple of projects. So I would definitely chat her up about this. Another concern was we spoke about the IGFs and how there should be a balance and everything. Just a curious question, that how can this translate to the diabetic patients, the patients who have diabetes? Because as a clinician, I'm always concerned about the clinical aspect of whatever the discussions that's going on. So hypothetically speaking, if this does convert into a clinical study further on, what do you think, in your opinion, just your opinion, would be for the diabetic patients? How would this affect the diabetic patients?
You know, of course, metabolic syndrome is always connected, you know, with malignancies very often, yeah, and other complications. You know, IGF-1, another name for IGF-1 is somatomedin C. It is IGF-1 secreted from liver. You know, we have tissue IGF-1, and you know, if we don't have IGF-1 at all, then it is impaired. Mice are dying, mostly because they have underdeveloped muscles, and as you know, breathing is active, yeah, and you don't have, you know, in breast muscle, you know, they cannot breathe and dying. This means tissue, you know, IGF-1 is important for development, but circulating in peripheral blood, somatomedin C or IGF-1 is problematic. And you know, and it is stimulated also by growth hormone, you know, the secretion. This means these two, at least, you know, factors are going hand in hand as culprit, you know, in damage. Yeah?
Well, this is really great. Scott, you had a question?
Well, sir, when you asked whether additional VSELs could be given therapeutically, and the response that you gave, sir, was that when IGF-1 is given, it's been shown to be deleterious to shorten lifespan, to promote oncogenesis. Has anyone done any research on giving H19 and its downstream products?
There were such papers, you know, come from Korea. I can give you literature. No, there were such papers done by Korean group. And also, what is here very important, you know, which is here with IGF-1 and IGF-1 story, that as I mentioned, you know, if we give to mice IGF-1, if we give insulin, we deplete number of VSELs. But on the other hand, in this expansion strategy, to expand VSELs, we add not only nicotinamide, we add not only sex hormones I was showing, but also we add IGF-2. That they will proliferate, yeah. So this means you can tell like this: if it is low IGF-1 level in peripheral blood, VSELs are quiet, they sleep. If we stimulate with IGF-1, of course, they are used up very rapidly. And the risk is that they start to proliferate, get mutation that can grow cancer. Different scenarios you can make.
Well, this has been really fantastic. I know everybody's got to get to their next. So